Today's topic is polygenic inheritance. What's polygenic inheritance? Now, when a trait or a character can be controlled by more than one genes is called polygenic inheritance. It is discussed by a Punnett square. Now I represent here, that is a Negro. The allelic genes are capital A, and capital B, and the white, that is small a, small b, small b. When cross each other and produced the hybrid, that is capital A, capital B, small b. Now, it produces that four types of gametes, that is small a, small b, capital A, capital B, capital A, small b, small a, capital B, and small a, small b. Now, when these gametes, that is male or females, meet with each other and mix the Punnett square, I represent here, that is male gamete, female gamete. Those are capital A, capital B, capital A, small b, small a, capital B, small a, small b. Same capital A, capital B, capital A, small b, small a, capital B, and small a, small b. Now it is capital A, capital A, capital B, capital B, capital A, capital B, small b, capital A, small a, capital B, capital B, capital A, small a, capital B, small b. Now, capital A, capital B, small b, capital A, small b, capital A, small a, capital B, small b, capital A, small a, small b, small b. Here, capital A, small a, capital B, capital B, capital A, small a, capital B, small b, small a, capital B, small a, capital B, small b. Here, capital A, small a, capital B, small b, capital A, small a, small b, small a, capital B, small b, small a, small b, small b. Now, when I repeat the genotypic ratio of this, that is Negro, the total number of Negro is 1/6. Why 1/6? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Only one part of 16 against 16, 1/6. That is black. Black is 4/16. That is black intermediate. That is 6/16. And the black, light black equal to 4/16, and white only 1/16. That is the ratio of 1:4:6 4:1. So now I discuss about the pleiotropy after the polygenic inheritance. What is pleiotropy? Pleiotropy is the other genetic phenomenon. Pleiotropy. What is pleiotropy? Pleiotropy means when a single gene can exhibit multiple phenotypic expressions. Such a gene is called pleiotropic gene. That is the HPS gene, that is HB beta gene. What is this gene? Is this type of genes can responsible for the disease of a sickle cell anemia. Sickle cell anemia. Sickle means the equipments cutting paddy. That is the when the RBC of humans can hold the shape of this type is called is produced the disease of anemia is called the sickle cell anemia. Sickle cell anemia is controlled by the genes that is HB beta S. These genes protect from the dangerous disease, that is malaria, which is the parasite oriented, that is Plasmodium vivax or Plasmodium malariae oriented diseases. Those type of Plasmodium can eat only the globin part of the RBC of human bloods. So at this stage the sickle type of RBC is not the automatic choice of Plasmodium parasites. So it is the another important of the protection of malaria. It is the examples of pleiotropy. Now we learn about the chromosomal part. They carry on the inheritance. Chromosome theory on inheritance. Chromosomal theory on inheritance. Inheritance. What is chromosome? Chromosomes is the fiber or rod-shaped structure of materials found only in the nucleus of eukaryotic cells, which carried out the genetic materials from one generation to next generations. Now, automatically the structure of chromosomes are here, that is this type of rod-like structure or fiber-like structure is called chromosome. What is the level of this structure? The bulb-like structure is called a satellite. Yeah. All the chromosomes can be divided by two same and similar shape and size of fiber-like structure, that is called chromatid. Each of the structure is called chromonimata. Now, here it is also chromatid and it also chromonimata. There are the so contracted part of chromosome is present, that is called the constriction. Here it is the constriction, and it is the another constriction. That is primary constriction. That is secondary constriction. In the primary constrictions, in which place only the chromatid or chromonimata is found, it is called the centromere. And the platelet structure covered with whole part of centromere is called kinetochore. And there are 4 nodules like structure made the kinetochore, that is, it's called centromeric chromomer. Now, another constriction is called secondary constriction in the interphase. In the interphase, these chromatids are joined with nucleolus, but in the metaphase stage or the telophase stage, this part produces the nucleolus, that is called nucleolar producing part, that is NOR, nucleolar organizing region. Now, there are so many parts found in the physical structure of chromosome. Now, it is the centromere. According to the presence of centromere, the number of centromere, chromosomes are divided into 4 types. No centromere found is called acentric. One centromere found called monocentric. Two centromeres found dicentric, and more than 2 centromere is found, it's called polycentric chromosome. According to the position of centromere, what is the position of centromere? The not middle or not ending part, here I represent the positions of centromere. But the after the centromere, all of the part of chromosomes is called telomere. According to the positions of centromere, the chromosomes can be separated 4 types. That is, when the centromere positions in the middle part, that is metacentric chromosome, that is the submetacentric, that is acrocentric, and the end portions where the centromere is present at the chromosome is called telocentric chromosome. So there are 8 types of chromosomes can be classified. Only the positions and number of chromosomes, centromeres, that is acentric, monocentric, dicentric, polycentric, and metacentric, submetacentric, acrocentric, and telocentric chromosome. Now there are 2 types of chromosomes, are autosomes and sex chromosomes. Autosomes and sex chromosome. What is autosomes? Those type of chromosomes can control only the somatic features of our bodies, as called the autosomes. Besides autosomes, all of the chromosomes is called sex chromosomes. Those chromosomes control the sex linked features of our body. It's called sex chromosomes. In human, there are 44 autosomes are present, that is 44A, and there are 2 in male, that is XY, in female, that is XX chromosomes. Total 44 plus 2 equal to 46 chromosomes present in all the cells of our body, but the sperm cell or ovum cell, that is the gamete produced respectively the male and female, where the number of chromosomes is haploid, that is 23. So haploid set of chromosomes is called the genome. Now we learn about the chemical structure of chromosomes. Chemical structure of chromosome. There are 2 separate parts makes the chromosome, that is nucleic acid and nucleic protein. There are 2 important protein makes the nucleic protein of the chromosome, that is basic and that is acidic. Basic proteins mainly called the histone type of protein, histone typic, and acidic proteins called the non-histone typic. Histone typic proteins are made by this named amino acids. Those are histone 1, histone 2A, histone 2B, and histone 3 and histone 4. Non-histone typic are made by the amino acids, that is tryptophan, tyrosine, etc. Non-histone typic nucleic proteins, that is H1. What is the component of H1? H1, the molecular weight of H1 equal to 2300 Daltons. The amount of lysine is huge. Huge amount of lysine makes the histone 1 proteins. Lysine is the amino acids. Now, H2A, the molecular weight of H2A is 1400 Dalton. The lysine is greater than arginine. Here, the lysine amino acids and arginine amino acids, both two type of amino acids present in histone 2A, but lysines, the amount of lysines is greater than the arginine. H2B, the molecular weight of H2B is 1300 Dalton. Here, lysine is present, but minimal, so minimal, greater than arginine, but less than the amount of lysine of H2A. But H3, where it is the molecular weight 11,300. Sorry, 15,000. 15,300 Dalton. The arginine is greater than lysine here, and molecular weight of histone 4 H4, that is molecular weight is 11,300 Dalton, where arginine is so huge. That is the features of amino acids presence found in histone typic protein of nucleic protein. There are two types of nucleic acid found in chromosome, that is DNA and RNA. DNA made up phosphate, deoxyribose, sugar, and deoxyribonucleic sugar and nitrogen base. Here RNA also phosphate, ribose sugar and N2 base. Nitrogen base is two type, purine and pyrimidine. Here nitrogen is two types, purine and pyrimidine. Purine are adenine and guanine. Pyrimidine in DNA, thymine and cytosine. Adenine bonded with thymine. There are two bonded of hydrogen ions and three bonded of hydrogen ions in guanine packed with cytosine. Here in RNA there are purine is adenine and guanine, but pyrimidine is uracil and cytosine. So adenine packed with uracils, the bonding of hydrogen double bond, and guanine hydrogen twin bond. So all of the chemical component of chromosomes are displayed here. Thank you.